Welcome to Chapter 3. First, I'd like you to take a second and move your eyes across the honeycomb pattern at the top. And what you'll probably notice is that not only are you seeing a sphere and then kind of a flared column and then a background, but as you move your eyes around, it seems like parts of the parts of the scene that you aren't looking at seem to be moving. They aren't moving. It's a visual illusion. And this is something that allows us to understand a little bit more fully how our visual system actually works. When we see ourselves detecting motion that's not there, or detecting color that is not present, or shapes that are incorrect, those kinds of things, it gives us a lot of insight into how the visual system works. So I just thought it would be fun to show you this. Every year there's a competition to see if anyone can come up with a new visual illusion, and this is one that was designed a few years ago. So I thought I'd share that with you for fun, and then we'll move on to the actual subject. All right. So we're going to be talking about visual perception in this chapter, which is going to cause us to go into a little bit of the biology of the visual system. We're going to talk about form perception, how we make sense of the visual field and determine what is the part we should be paying attention to and what part we should be ignoring. We're going to talk a little bit about how our visual system maintains constancy in its perceptions, and then we'll wrap it up with a discussion of how we perceive depth. All right, let's get going. All right, here's a nice diagram of the eye and the components that are important. So this eyeball is obviously looking up, and the outer area is your cornea. That's the see-through part that holds behind it a chamber of liquid, basically. It's called aqueous humor. It's a liquidy humor that's much more fluid-like, much more watery than the vitreous humor that's going to be behind the lens. So behind the cornea is a liquid chamber, and then behind the cornea is your iris. The iris is the part of your eye that has the color. So some of us have blue irises, or green irises, or gray, or brown, or something in between all of those, or lavender, or different colors are possible, right? The iris is created by strands of reflective fibers, and the color that is reflected off of those fibers determines what color your eyes appear. So we've got your iris, and the iris surrounds the pupil. The iris is actually what controls the size of your pupil and determines how much light gets let into your eyeball. So in very bright light, your pupil will be very small, so as not to let too much light in. And then in low light, your pupil will get really dilated, really large, so that you can allow more light in, so you can see better. The light first passes through the lens, which needs to accommodate the distance of the light source. And so when we're younger, usually our lens is nice and flexible, and it can really accommodate well from very close up to very distant objects. It can bend and accommodate that light. As we get older, the lens gets a little bit more rigid, and so it doesn't bend as much. And so older people tend to develop farsightedness, where they can see things at a distance pretty well, but they need reading glasses. It's called presbyopia because presby means old. So as our eyes age, we're more likely to need glasses to read. So the lens accommodates the light and decides how to focus it on the back of the eye. That light then has to pass through that big chamber between the lens and the back of the eye, and it's full of something called vitreous humor. And vitreous humor is sort of the consistency of Jell-O, or if you're from a British-speaking country, that I think you'd just call it jelly. So it's kind of got that kind of bouncy, not liquidy, but kind of resistant kind of texture to it, and that fills the eyeball. So the light has to pass through that kind of not purely clear vitreous humor to get to the back of the eye. What we're going to spend our time talking about in this section is the very back of the eye, what's called the retina. In this diagram, it's drawn in yellow, and it kind of gives the impression that it's only in two dimensions, but the retina covers the entire back of the eyeball, from the top to the bottom to the sides. It's covered in retina. There's a specific area of the retina that is called the macula, and then right dead center in the macula is the fovea. And the fovea is the part of your retina that is going to collect the information from the object that you are fixating on, the thing that you're really looking at. That information lands on your fovea. Other big structures to look at here, they made sure to include all the blood vessels and things that are in your eyeball. And you see that they all exit in one place. They all come in and they all exit in that one place. And that's in the middle of the optic nerve. So all of the light sensing cells that are in your retina are all going to exit your eyeball in that one spot, as well as the vessels that you have in your that need to nourish your eyeball. And that's going to produce what's called an optic disc, which is the part of your eyeball that actually doesn't collect any information. So it creates a blind spot. So we each have a blind spot in each of our eyeballs. The good news is, because you have two eyeballs that are roughly pointed in the same direction, the blind spot in one eyeball is compensated for by the other eyeball. And then its blind spot is compensated by the other eyeball. So you don't usually have a sensation of a blind spot unless you very deliberately take some steps to make that blind spot obvious to you, like cover one eye, fixate on something, and then you'll notice that part of your visual field actually isn't being collected. But most of us don't do that in our everyday lives, and we normally don't even notice we have a blind spot. All right, so those are the main structures. Like I said, we're going to talk about the retina. So let's go ahead and zoom in on that. So the retina, like I said, is the back lining of the eyeball. And it's made up of two light receiving types of cells, the rods and the cones. And if you took Psych 100, I'm pretty sure your teacher probably taught you about this. If you took biology, you might have learned about this. These different kinds of what we call photoreceptors, light collecting cells. So rods, their advantage, their special skill is that even in low levels of light, they are still sensitive. So at dusk or after dark, your rods are going to be collecting most of the light that you are able to process. They have lower acuity. You're not going to be able to see as clearly in dusk or dark conditions as you could in bright light, because your rods don't collect very much information. They give you good information about motion or that something is there, but you may have difficulty reading in low light, for example, because your rods aren't very, they don't have great acuity. They are colorblind. They only see shades of black and white. So black, white, gray, that's all they can really distinguish. So they give us a lot of information about contours and shadows and things like that. Black font against a white paper, something like that. That's going to be your cones. Your rods. There are no rods in your fovea. The point of fixation does not have those black and white receptors. So you're going to be getting all of the information about contours or like outlining on something that's in black and things like that. You're going to tend to get that from your rods that are outside of the fovea. Now your cones would be the other kind of light receptor, and they don't function very well in dim light. Your cones need good solid light in order for them to function correctly. They tend to have greater acuity. They see more detail. They collect more information. They're color sensitive. So your cones are going to help you to see that the things that are on the screen right now are different colors, and maybe to even help you to figure out which exact colors those are. Some of us have better color sensitivity than others of us do, but they are the part of our eyeball that gives us that color information. They're mostly congregated around the fovea. We really don't have cones out in the periphery. So our peripheral vision tends to be black and white. It's weird though, because our brains don't perceive it as black and white. Our brains figure it out. Maybe you turn your eyes and go collect a little bit more color information, and that'll help fill in the blanks about what color thing you're actually detecting in your periphery. So your visual system is designed very efficiently, where it's like, okay, well, I'll have a lot of rods out here in the peripheral vision. It'll get the blacks and the whites and the grays and these kinds of big, gross contour kinds of information. And then in the point of fixation, I'm going to focus in on light. I'm going to focus in on color. And I'm going to get good, clear information in the point of fixation. And you might notice that if you're ever reading, that the words that you're looking at the moment are in focus, and the ones that surround it that you aren't paying attention to, they tend to be a little blurry. And that's because the rod is picking up that information. And you don't really need that information to be in great focus because you're not paying attention to it right now. You're only paying attention to the ones that you're actually moving your eyes and trying to look at. So it's okay that other things are out of focus. What our rods are great at is helping us to notice if there's any motion. So if something moves out of the corner of our eye, we turn our eyes and we look at it. If we see an object out of the corner of our eye, it may be just a sort of shapeless, imprecise, color's not there kind of form, but we know, there's something that is over there, I'm going to look. Right, and now I turn my eyes on it and get more information. So it's a really efficient system. So here on the left is an actual, electron microscope image of the retina, and you can see the rods and the cones. This is in a part of the retina where there are cones and rods in the same area. And they've colorized for us the cones in a shade of green and the rods in a shade of mustardy brown. I don't know what color that is. And you can also see the support structure beneath them. It looks almost like they're on moss that's been colored pink, or maybe coral, right? It's on a coral reef or something. It's weird how spongy that structure is that is holding on, that the rods and the cones are nestled in. But that's, zoomed in, 1,000 times, right, for you to be able to see that kind of detail. Now on the right side, it's showing us the relative number of fovea of rods and cones in the fovea and in the non-fovea areas. So you see that there's, a peak of rods near the fovea, right, but not in the fovea. And you see a peak of cones in the fovea. And then you see that one gap where that's the blind spot where no photoreceptors are collecting information. So we see that there's some overlap of rods and cones in certain areas, but there's very much specificity in the eyeball about which kind is located where. And of course, the function of these photoreceptors is to collect light energy that's being reflected off of objects. We can only see a narrow band of the whole spectrum of light waves that are out there in the world. If you look at the very bottom, it shows you there's all these different kinds of light rays, and we can see that little narrow band that's broken up into the colors of the rainbow. And that's the only part that we can actually see. We can't see broadcast radio or gamma rays, but we can see this one little narrow band called visible light. All right, now, how do our rods and cones convert into something that our brain can actually visualize? We call this visual phototransduction. So photo refers to light. Transduction means translating that from these little elements into something that your brain can actually understand. So the eye, with its photoreceptors, and also with the bipolar cells and the ganglion cells. So what you were seeing in this picture is that actually the rods and cones are at the deepest layer of the retina. So the light comes in through your pupil, passes through the lens, passes through the vitreous humor, passes through all these layers of the retina that you're seeing in front of you to finally be collected by the rods and cones that are at the very back part. And then once that information has been collected, it moves back through those red ones, which are called the bipolar cells, then through the white ones, which are called ganglion cells, and ultimately to the optic nerve. So just like we were talking before about how information crisscrosses from your right hand to your left hemisphere and like that, there's a lot of structures in our brain where the information has to go probably farther than you think would be logical before it actually starts to move back in the correct direction. And the eyeball is one of those examples. So it goes from these photoreceptors through the bipolar to the ganglion cells to the optic nerve. And the optic nerve is going to conduct this information up to the thalamus. And we talked about this before, how all sensory information first gets transferred to the thalamus. And the thalamus decides where to send the information for further processing. So if you think about the thalamus as a nice switchboard where, I don't know, switchboard is an old-timey thing to say. I'll say router. If you think about the thalamus like a router where all incoming information goes there first, and then it sends it to this device or that device or whatever within the household, that would make a lot of sense to think of that kind of model, right? That it's sort of like that. From the thalamus, it's going to get cast back to the occipital lobe of our brain. Remember, we talked about that before. That's the very back of your brain, which contains your primary visual cortex. So if we look again at a picture of the brain, we're seeing at the top, we're seeing it from as if we were on the inside of the brain looking out. And then on the bottom, it's like we're looking from the outside of the brain towards the center. And so we see that each hemisphere has an occipital lobe that has a primary visual cortex for processing visual information. Something that's really important to know about how the primary visual cortex operates is that it displays what's called lateral inhibition. So here's a little bit of a diagram. This is from our textbook, so there's more explanation exactly about this diagram in the textbook. But the basic idea is that as information comes in, so for example, with a bright physical stimulus on the left-hand side, the cells are trying to send the information the vertical way. So from where it says cell A, it wants to go down its axon and send it to its destination, which is at the bottom of that diagram, to the brain. So it wants to be passing it down in that vertical way. There's what we call lateral inhibition. See those little green areas that connect the red neurons? Those are connections that prevent those two neurons from telling each other about something that both of them are already trying to process anyway. So that lateral inhibition allows the information to flow from the top in this picture to the bottom in this picture without sort of disseminating it out laterally and telling the neighboring cells, who in this case already know, or maybe in a case where it's a low light level, where maybe I don't need to be passing along this information to my neighboring cell at all, because that one, it's not that bright of a light. You can sleep. It's okay. I'll take care of it. So we don't want to have information shared side to side. We want it just be being shared downstream to where we're trying to project it. And so that's one of the really interesting facts about how the visual cortex actually operates. All right, so that gets us through sort of how information, at least in the visual sense, gets collected. In the next video, let's go ahead and make a nice distinction between sensation and perception, and then move on with some facts about that. So I'll see you in the next video.